Good morning, everyone. So my name is Mathias Svahn. I'm CEO and co-founder of NextCell Pharma, and we develop mesenchymal stromal cell for autoimmune diseases. Our primary indication is type 1 diabetes. We are a listed company on NASDAQ First North, so this is our disclaimer. You can read our quarterly reports every quarter. It's very exhausting to be on First Growth North market, so if you don't have to go there, maybe you should wait a while. An executive overview. So we are developing an allogeneic off-the-shelf cell therapy product to treat hyperinflammation. We have treated to date over 90 patients in 5 different clinical trials. We have shown that a single infusion have long-lasting efficacy, even though the cells are not engrafting. We see effect over more than 5 years. We know that we can give repeated doses without triggering an immune reaction. So this is truly an allogeneic cell therapy that is off the shelf. We store it in liquid nitrogen, and we have a shelf life of over 3 years. So you can, when needed, you can just take out a bag, thaw it, and give it to any patient. Our manufacturing process is patent protected, and I will show you a little bit more of our core technology. So we are now we have data from phase 2 and are looking for global or regional partners to develop this therapy and bring it all the way to patients. So our company was founded in 2014. We founded it as a stem cell bank to really build the infrastructure and the know-how about the logistics of these cells, taking care of them, and also securing high-quality starting material. So we built our lab on the 8th floor in this house. You have to take the D elevator to reach us. You cannot reach us from this floor, which is very inconvenient. So it's under the fire escape staircase where it's a smoking area. There is our main entrance. You can also choose to find it through level 3. Then you can walk within the whole body of the house. But we built clean rooms there to establish our tissue establishment and biobank, and launched the service of private stem cell banking in August 2015. Then we developed the company to also develop therapies based on mesenchymal stromal cells from umbilical cord tissue in 2016. And in 2018, we treated the first patient. And then we have been moving pretty fast, I think. So I'm one of the co-founders. Lindsay Davis, that most of you know, she is our CSO. And Patrick Fagerholm, who we got from Sprint Bio, thank you very much. He is now leaving us because he was the interim solution. He stayed 4 years, so something was right in the working environment at least. But we're now seeking a new one. And then we have our chief medical officer, Edvard Smith, or Ted, that most of you know him as. So our primary indication. Type 1 diabetes, as you know, is an autoimmune chronic disease. It's very heterogeneous, and we are not fully understanding the mechanism why this disease is developing in most cases, and it varies. We have been treating with insulin for the last 100 years, but there's no real treatment of the underlying disease. It's just handling the symptom. There are a lot of different immune cells involved in the degradation or the degeneration of beta cells. And there have been some trials with immunosuppressive drugs where you use typically antibodies to knock out the specific signaling pathway. And this can be more or less efficient, but the side effects are severe, and the long-term complications might be not acceptable when it comes to children who then will have a suppressed immune system for many years. Our approach is targeting the same sites, but it's more of fine-tuning the immune system. So these cells will not knock out a single pathway. They will influence all the pathways through different signaling mechanisms, and then generating an overall effect that is very potent, but with no side effects. The whole core of our product is how we pick and choose the optimal cells. We know a lot of the biology of how these cells work, and we were assessing our manufacturing process while developing this drug and realized that we have developed a whole panel of different functional and potency assays to really measure the immunomodulatory effect that these cells have. So we formulated this as a patent to pick and choose optimal cells using a selection algorithm where we make an overall assessment of multiple assays. So we have a theory of a fixed set of more general assays, and then we can pick disease-specific or therapeutic area-specific assays into that to really optimize the way we select the cells for the specific treatment. The cells are produced only for 3 passages to prevent them being exhausted. And in the last step, we pool cells from multiple donors. So we pool cells from 5 donors to generate large batches with extremely low batch-to-batch variability. It's also a very important stimulation step for having these cells being primed for immunomodulation. Then we aliquot them in bags of 100 million cells per bag, and these cells are then truly off the shelf. After you thaw them, it takes 3 minutes. They are connected to an infusion bag, and you invert the bag like a time glass and give it as an intravenous infusion over 20 minutes. For children, that's 1 bag. For adults, it's 2 bags, so therefore it's 40 minutes. So it's extremely simple treatment. It's a fixed dose, and this batch size, we can now produce hundreds of doses, but we could expand this for even larger batches. So to exemplify how the efficacy looks like for treatment with a single infusion, I'm just going to present the phase 2 data from our adult clinical trial. We treat the patients within 2 years after diagnosis in order to save as much insulin production as possible. The primary efficacy endpoint is measuring insulin production before treatment and then 1 year after. So they are getting a single infusion with 200 million cells, and we follow them over time. And our primary endpoint was statistically significant at 1 year, where treated patients lost on average 10%, whereas placebo lost 50%. But the most impressive part is that it's really a disease-altering therapy with a single infusion, using cells that will be gone within 72 hours. And after 5 years, our treated patients still have more than 50% of their insulin, which is then more than the placebo group had after 1 year. These patients are having insulin pumps and continuous glucose measurements, and they are basically almost in range. They are doing very well in their diabetes handling. But there are differences between having exogenous or endogenous insulin. And we can see other broader health benefits by preserving your insulin in BMI. So the patients treated with placebo, they gained weight over time. In average, 3.5 units, which on me would be 15 kilos, so substantial gain. The ProTrans-treated patients were still at the same weight after 5 years. This was also reflected in blood pressure. So really a disease-altering therapy with a single infusion. In addition to this, yes, we can give this multiple doses. Even though we give a pooled product with 5 different donors, we don't see any HLA antibodies de novo. We have treated some patients with two doses, first one and then waiting 1 year and then another one. So they've been exposed to 10 different donors. And even though they had pre-existing HLA antibodies, they were not sensitized. So we know we can give this repeatedly. We have treated now patients down to 7 years old in our pediatric trial. It's running in Uppsala, Linköping, and Malmö-Lund. So 66 patients in that trial, and we are now in the follow-up of these patients. We also done 2 clinical trials in COVID. One in Canada was sponsored by McGill University, and one in Örebro, which was later expanded to more general viral-induced severe pneumonia. Yeah, but I didn't bring any slides for that because it would be too many. So what we have looked at to develop is really an industrial way of making cell therapy available for patients. We have produced or developed a drug that is effective and safe with a single infusion. So that is very much appreciated by the patients. For the doctor, this is an off-the-shelf product. It's very straightforward, no post-processing of the product, just a fixed dose infusion. It is a cost-efficient treatment in the sense that it's very simple procedure. We don't see any side effects. You don't have to be in a university hospital to give this therapy. And when it comes to manufacturing, it's patent protected, and given the way we are analyzing our cells for functionality and potency in manufacturing process, it's very robust. So we are now looking for out-licensing or co-development for different areas. We are also product developing for other diseases, and we have announced that we will have a global collaboration with Fujifilm Biosciences for selling our mesenchymal cells for research purposes to start with. NextCell has now also 2 daughter companies. So we have a broader knowledge base than only just mesenchymal stromal cells. From Cellaviva, we have a SOHO tissue establishment, SOHO entity it's called now. We have long-term biobanking and diagnostic services, but we also help other companies with HLA, infectious disease screening or genetic profiling. And we are also distributors for companies, foreign companies that have tissue products that they want to import to Sweden, so we can distribute them to clinics. And our most recent company is Qvons, which is a QC analytics company, really focusing on ATMP products for the reason that NextCell, we were looking for these analyses ourselves. And our core business will be endotoxin, mycoplasma, and sterility. But we will expand the portfolio. Yeah, that was my little pitch. And I would be more than happy to take questions or invite you for a tour whenever.